To get started on your virtual labs, you are going to need to have to read through the introduction for the lab in your lab manual. There's important information and background information for you to understand your flowers and trees. The lab manual is set up in a step-by-step process. So as you read the steps, you can complete them on your computer software. We have done a video to help give you some guidance in how to proceed in some of the pitfalls you might run into as you're working through your software. But it's going to be imperative that you read through the introduction and that you read the directions carefully and answer the questions in your lab manual as you proceed through. An important part is that you do not need to complete the entire lab in one sitting. All you need to do is finish out each exercise, and then you can close out your software. So for instance, you come in and you get started and you complete exercise 1 and you go to exercise 2. As long as you finish all of the steps in exercise 2, you can close out that software. And you can start tomorrow and begin exercise 3. If you close out your software in the middle of an exercise that you have not completed, you'll need to start that exercise from the beginning. The computer will randomly generate these trees, so you're not guaranteed to get the same tree back if you try to pick up in the middle of an exercise. So please remember, finish each exercise, then you can close out your software. You will not need to do Flower Challenge 2 at the very end of this lab. That's a much more complicated phylogenetic tree to try to reconstruct. Also, you don't need to go through and look at why are there so many species of columbine on your own. This is going to be your introduction and simple directions for how to complete the virtual lab called Flowers and Trees. Your instructor will post on your Blackboard site a link where you can download this Symbio virtual lab software onto your own computer. It will not cost you anything to download the software. The site is completely safe to download off of. And when you click on that link, you will be brought to a website that gives you the option for downloading onto a Mac computer or one that has Windows. When you click on either of those links, it will bring you through the steps that you need in order to download that software onto your computer. Once it's on your computer, you will see an icon that looks like this, Symbio Virtual Labs. And when you click on that icon, it will bring up a screen that looks like this. Our Flowers and Trees lab is going to be this one under Evo Beaker Labs. And when you click on that, it will refresh and bring you to a screen that looks like this. We're going to be working with the flower columbine in this lab. Columbines are annual plants, meaning in one growing season, they grow from a seed, produce the plants, produce flowers, which is how plants reproduce sexually. That flower gets pollinated and it produces seeds. Those seeds get distributed for next year's growth. All of its growth takes place in one growing season. So each year in our simulations that we look at is one generation in the life of this columbine plant. We're going to focus in this lab on changes that can take place in the flowers. Changes can also occur in the leaves, the stems, the roots. Over here, you have different mountain peaks that are going to have columbine flowers growing on them. The first part of your lab is just to get used to the different parts of the flower and what can change. So when you click on one of these flowers, you'll get a screen that looks like this. I move mine in front of that because the changes are going to occur on this little flower here. So you can change the color of the anthers from white to yellow. And now look, those are the anther part of your flower. Your petal tips can go from round to pointy. Petal color can go from light to dark. So we can make all sorts of changes in these flowers. All of these changes are the results of mutations that occur. Changes in the DNA that this flower has. Keep in mind the name of this lab is Flowers and Trees. They're not trees like maple and oak trees. They're actually evolutionary trees, looking at how related two populations are. And so we're going to track changes over time, and we're going to see which populations are more closely related to each other. So one thing we can do that you'll be looking at is we can grow trees. Your controls are all down here. You have a go or start, you have a stop, you can reset, and then you can do it step by step. So watch what happens when I hit the go button. My tree starts to grow. Now notice, my tree started here with peak 7. And peak 7 went along and kept reproducing and growing year after year. Here's our years, 0, 50. Okay, we're at almost 100 years. And look at what happened here. Our population split. Seeds from peak 7 were carried over to peak 1. How? Wind could carry them. Animals, birds could carry them. People could carry them. Hikers could pick up those seeds that get stuck to their boots or their clothing. And those seeds get deposited on a new mountain. And now you have another population of columbine. Something else has happened here. We've picked up a change or a mutation in the anthers in that flower. That mutation only occurred on peak 7, not on peak 1. And so we see a change that has taken place in different colors on our mountain peak. Let's run our simulation a little bit further. Now we're up to 3 peaks of flowers. And look, there's the color change for our anthers. And if we track it all the way down, there's where it took place. So peak 7 and peak 3 have changes in their anther color. Peak 1 didn't, because peak 1 was populated before that mutation took place. So now peak 7 and seeds from peak 7 have gone and populated mountain peak 3. So we have 3 mountain peaks here, all tracing back to our original ancestor all the way back here. When we look at how related peaks or populations are, it's all going to go back to the amount of time that has occurred between each peak and their most recent common ancestors. And that's what these circles represent. So if we look at peak 7 and we look at peak 3, we come down vertically in our time, and then we go across horizontally. This is the most recent common ancestor between peak 7 and peak 3. And it lived back here about time 175 years. So from 250 to 175 years, that's about a 75-year difference. But now, if we're looking for the most recent common ancestor between peak 7 and peak 1, we have to come all the way down until we go horizontal here. We have to go all the way back to year 75. So the most recent common ancestor between peak 7 and peak 3 was 75 years ago. The most recent common ancestor between peak 7 and peak 1 was 175 years ago. And our original ancestor is all the way back here at time 0, 250 years ago. The hard concept to understand is peak 7 and peak 3 are more closely related to each other because they share a most recent common ancestor. Peak 7 and peak 1, we have to come all the way down here to find their most recent common ancestor. So when we look at our 3 peaks, peak 7 is more closely related to peak 3. What you have to understand is these ancestors are no longer alive. They all occurred in the past. Peak 7, to get from peak 7 and the flowers that are alive today to the original ancestor in the past, we have to go 250 years. To get from peak 3 to the original ancestor, it's also 250 years. And to get from peak 1 to the original ancestor is also 250 years. All 3 of these peaks are equally related to the original ancestor because the same amount of time has passed to get from their current population to the ancestor. So all of them are equally related to the original ancestor. But remember, peak 7 and peak 3 are most closely related to each other because they share that most recent common ancestor. And you are going to work through some exercises, exercise 2, exercise 3, that's going to look at how these populations are related, and it's going to translate that to you and your siblings, if you have any, and your family tree. Keep in mind, when we look at our family and our family tree, we're looking at generations. So I'm the youngest of 5. I have an older sister. She's still in my same generation. My older sister, although she's older than I am, she is not my ancestor. The ancestors that we share go back to the next generation, our parents. So we would share our most recent common ancestor as our parents. If we went back a further generation, then we would go back to our grandparents. So when they're asking you about how closely related you are to your sister or your cousin, keep in mind generations for humans are longer than the 1 year we see in our columbine flowers. You will also be asked to look at your phylogenetic or evolutionary tree and see what happens when you move things around. We can slide them back and forth. It doesn't change anything. It doesn't change the time frame. It doesn't change who's most closely related to each other. All it changes is the order that we see things on. We have a more complicated evolutionary tree up right now, where we have flowers on peak 4, peak 3, peak 6, and peak 5. And we've had changes occur in peak 3 over here that's going to affect peaks 5 and 6, and this one down here that affected all of peak 3, peak 6, and peak 5. When we look at that tree and you're asked the question, which peak was colonized first, our instinct is to go, oh, peak 4. It's off here by itself. No, Just by giving a tree that looks like this, you cannot tell which peak was colonized first. We could start at peak 3 here, and if this is peak 3, it would have split to peak 4 first. And then if this is peak 3 here, it's gonna split maybe to peak 6. And then peak 6 would split to peak 5. Or this could be peak 5. And if it was peak 5, it split off to peak 4. Then peak 5 split to peak 3. And then peak 5 split to peak 6. You cannot tell just by looking at an evolutionary tree which peak was colonized first. Any one of these peaks could have been the original peak that was colonized and led to a tree that looks just like this. The other complicated part is which peak is most closely related to the original ancestor. Again, our inclination is to say peak 4, it's off by itself. It doesn't have the changes that these other peaks have accumulated. But remember, relatedness goes to time past. Peak 4 to the original ancestor, 350 years have passed. Peak 3 to the original ancestor, 350 years have passed. Peak 6 to the original, 350 years. And peak 5 to the original, also 350 years. All 4 of these peaks are equally related to the original ancestor because the same amount of time has passed to get to that original ancestor. Now, we can look at individual peaks. Peak 6 and peak 5 are more closely related to each other because they share a more recent common ancestor. And then peaks 5 and peak 6 are next related to peak 3 because here's that most recent common ancestor. And finally, those peaks are then related to peak 4. So you have to keep in mind individual peaks can be most closely related to each other depending on where the most recent common ancestor is. But when we look at all of these peaks to the original, they are all equally related to the original. And again, it doesn't matter when you do the flipping out exercise. It doesn't matter, okay, in what order we move them around. That doesn't change anything. It doesn't change when they have accumulated mutations. It doesn't change the time. It doesn't change who's most closely related to each other. All it changes is how we view or look at that phylogenetic tree. Keep all of those things in mind when you get to exercise 5, which is your tree building challenge. You are going to have to construct a phylogenetic tree that looks just like the one in your lab manual on page 80. However, you cannot start at peak 4. You have to start at a peak other than 4. So when you go to that tree building challenge, okay, and you get here, you have to start at a peak other, okay, than peak 1. You can start at peak 2, 3, or 4. And so here you go. I'll help you out with the first step. If this is our tree we're trying to make, and this is peak 4, we're gonna let our simulation run for a few steps, and we're gonna stop it. Our first break, if this is peak 4, our first split has to be to peak 1. We will take a flower from peak 4, and we will drag it over to peak 1. And now we have our 2 peaks with flowers. We're gonna let it run for a little bit, and we're gonna stop it. Now, in our lab manual, peak 1 is off here on the left. So all we're gonna do is swap places. I've started the tree building challenge for you. You have to complete the tree building challenge and make it look just like what's in your book. So you need to populate peak 2 and peak 3 and have it look just like what's in your book. Okay, now we're on to exercise 6, reconstructing trees. You will only need to complete through flower challenge 1. You'll get a screen that looks like this, and you'll need to run your simulation, and you'll see all of your mountains start light up. And now all of them have been populated, so we're going to stop our simulation. We have columbine flowers on 4 peaks, and we're going to drag the flower into our notebook section here. So with your mouse, take one of the flowers, drag it, and drop it in. So there's peak 1. There's peak 3. Peak 2. And finally, peak 4. And now I can close that section so I have more room to work. Now notice, we have different colors on our petals, different shapes, different colors on our anthers and spurs. What we're going to do is try to group these 4 flowers from our 4 different peaks with similar traits that they exhibit. So let's start with our spurs down here. Peak 3 and peak 4 both have blue colored spurs. Our petals, peak 1 has blue petals, and so does peak 3. Peak 4 has pointed petals. None of our other peaks have those. So what we're going to start to do is we're going to use our little pencil tool, and we're going to draw circles around the peaks that share certain characteristics. So when you click on the pencil tool, you get different colors that you can use. So I'm going to get my red pencil, and I'm going to circle the peaks that all have blue colored petals. And now I'm going to get a different colored pencil, and I'm going to circle my 2 peaks that have the blue colored spurs to them, which is just, here we go, pencils are fun to use. We're going to circle peak 3 and peak 4. And you can do that with all the rest of your traits. I'm going to simplify this, and I'm going to show you then how you can make your phylogenetic tree. This tool down here is going to allow you to connect your traits together. And so when we start to look at, okay, peak 4 and peak 3 share, okay, those blue colored spurs that peak 1 and peak 2 don't, we're going to make a connection between them. And then peak 4 and peak 3 share those blue colored petals that peak 2 doesn't. And we can connect that. And then we can finally draw our connection all the way over to peak 2. Your solution will be revealed when you click that button. You can also you can drag, okay, these colors to correspond with your circles and your tree, but it gets a little crowded on your computer screen when you do that. And when you reveal, okay, you can see whether you were correct or not. And I obviously obviously did not do this correctly, but that's okay.